in Kingdom Monera, one other group is there, those are called as Cyanobacteria. So generally we call the Cyanobacteria are the Cyanophycean members. They belongs to Cyanophyceae, and other group is called as Mixophyceae. They belongs to both Cyanophyceae and the Mixophyceae, and generally these are considered with the blue-green algae. And here these Cyanobacteria have one special feature, that they may be either unicellular, multicellular, or colonial forms. And here when they are in the colonial form, they will be in the cocoid colony, and other one is called as dendroid colony. Actually, these Cyanobacteria are surrounded by some sort of mucilage sheath. So here in the cocoid family, the members which are present in the colony are surrounded by the mucilaginous sheath, but they are not connected to each other. But here the members which are present in the dendroid colony are connected with the help of mucilage which is present in that colony. So this is the way how cocoid colony is differentiated from the dendroid colony. And when it is a unicellular one, the single cell will perform all the functions. And in the multicellular structures, they contains more number of cells, and they are taking up the functions of the species for its survival and the life processes. If it is an filamentous form or multicellular structure, if it is multicellular, all the cells are arranged in the form of a filament. So we call it as a filamentous structure. This filamentous structure is surrounded by mucilage actually. Mucilage sheath is present. Some filaments which lack mucilage sheath, or without the mucilage sheath, the filamentous structure is called as trichome. So likewise, this type of organization will be there in the multicellular filamentous Cyanophycean members. Then here the cell wall is present around the cells, and this cell wall is helping in protecting the inner structure. Apart from the cell wall, they contains the cell membrane inside it. Inside the cell, that is lipoproteinaceous in its nature, and inside the cell membrane, the protoplasm is present. Here the protoplasm, which is having the cytoplasm along with the nuclear structure. So here the nuclear structure is not surrounded by the nuclear membrane, so we consider it as a prokaryotic in its nature. So it is having the circular, naked, double-stranded DNA structure, which is acting as a genetic material. And apart from this one, the protoplasm is differentiated into two parts. One is called as chromoplasm and centroplasm. Here centroplasm is a central colorless structure which contains other cell organelles, whereas chromoplasm is a peripheral and containing pigmented region. So here there contains a pigmented structure, that is chlorophyll A, carotenes, xanthophylls. Apart from this one, they contains phycobilins also. Particularly phycobilins are unique type of pigments which are present in Cyanobacteria. Here the phycobilins are of various types, like phycocyanin. The phycocyanin will provide blue color. The same way phycoerythrin. The phycoerythrin will provide red color, and allophycocyanin. This allophycocyanin will provide light blue color. Likewise, these are three types of phycobilins which are unique present in the cyanobacteria members. The genetic material, as we discussed here, it is in the form of double-stranded naked circular DNA. So we call it as a nucleoid, as like that of the eubacterial members. And apart from this one, as we say that pigments are there, these are present in special structures called as lamellosomes. Lamellosomes are the structures which contains the membranous structures which contains the pigments in it. So here the cytoplasm, as we had discussed, which contains the 70S type of ribosomes, and they lack other type of structures. So these lamellosomes, as they have this lamellar structures with pigments in them. So as they contains chlorophyll A and carotenes and xanthophylls, they are concerned with the process of photosynthesis. So that is they can perform photosynthesis and prepare their food materials. So this is the way how they are having this structure, and the cytoplasm will contains the large central vacuole. Large central vacuole is also present in them. So this is a way how the cells are exhibiting this type of special characteristic feature. They have reserve food materials in the form of cyanophycean granules and cyanophycean starch. These are two types of reserve food materials which are present in the members of cyanophycean. And apart from this one, they have ability to perform the functions in the anaerobic condition. That's why these are showing some sort of prokaryotic nature. There are some cyanobacteria which are useful for nitrogen fixation. Particularly the members like Anabina, nostoc. Such type of members are concerned with the symbiotic type of nitrogen fixation, or sometimes they may exist in the outer environment, that is some of them are acting as a free living and the nitrogen fixers. What is the significance despite of the prokaryotic nature and microscopic nature? What is the significance why they are able to perform this nitrogen fixation? Means they have a special enzyme called as nitrogenase enzyme, or we can call it as a dinitrogenase enzyme. So this is an enzyme which is responsible for the conversion of molecular nitrogen into the useful available form of nitrogen which is provided to the higher plants, in which the Anabina and nostoc are in the form of a symbiotic association. In cyanobacteria, if you see the filamentous structure, so this is a cellular structure, the filament. Here and there we can see some large sized structures. These large sized cells or the structures are called as a heterocysts, this one. And if you see the normal cells, so peripheral part will contain the pigments. The pigmented structures will be present. And it is an example for multicellular and filamentous structure. So here surrounding this filamentous structure, I haven't drawn the mucilage. Now we call it as a trichome. But once we draw this mucilaginous sheath around it, now it is a normal filamentous structure. So this is a heterocyst cell, and these are the normal cell with centroplasm and peripheral chromoplasm. So if the heterocystous forms are there, they have these heterocystous forms will take up the function of nitrogen fixation. Actually in the bacteria, they have leg hemoglobin which will protect the nitrogenase enzyme from the high concentration of oxygen. In the heterocystous form, this heterocyst, the special type of cells which are present in them, these heterocysts will protect the nitrogenase enzyme from high concentration of oxygen, because when oxygen concentration increases, the nitrogenase enzyme will become inactive. Once it become inactive, it cannot perform the, or it cannot help in the nitrogen fixation. That's the reason why the oxygen concentration should be maintained in such a manner that it should not cause harm to the activity of the nitrogenase enzyme. That function is taken up by the heterocyst. That is the reason why we consider the heterocysts are concerned or helping in the nitrogen fixation of the plants or in the cyanobacteria members. The cyanobacteria are differing from the eubacterial members by some characteristic features. Let us see how they are differing from the eubacteria. That is, the cyanobacteria will lack flagella, whereas we have seen in eubacteria they have the flagella, which is useful for the locomotion. And here these are photosynthetic, in oxygenic condition, whereas bacteria are non-oxygenic in their nature, whereas they have chlorophyll, carotenoids, and also the xanthophylls. Apart from that, they have the unique type of pigments called as a phycobilins, and they have cyanophacean starch in the form of reserve food materials, whereas in bacteria they have the glycogen in the form of the reserve food materials. Whereas, so this is a way how these structures are differentiating, that is cyanobacteria is differing from the normal eubacterial structures, and majority of them, or we can say some of them, are useful in nitrogen fixation also, but here the heterocyst structures, they have some special structures called as heterocysts, which are not present in the normal eubacterial members. And apart from this one, these are the structures which contains the covering like gelatinous sheath around them. So these are some of the characters where the cyanobacteria are showing some variation from that of the eubacterial members. There is a cyanobacteria called as Trichodesmium erythraeum. So this Trichodesmium erythraeum is responsible for red color of Red Sea. Red color of Red Sea is because of the pigment which is showing dominance when compared to the chlorophyll A molecule. And apart from this one, the type of reproduction that is done by this cyanobacteria is, so they participate in a process called as fragmentation. Apart from that one, they form small structures called as hormogonia. So hormogonia are the special small fragments which have been produced by them. These fragments will separate and they grow into individual ones. Apart from this one, they produce acinides. Acinides are the special structures which are having thick walled unicellular structures. So likewise, these are the various methods of reproductions which are participated and done by cyanobacteria members.